All right. So the other process is called extracellular junk, and so there's these beta amyloid proteins that that get produced, and so, for example, there are these two proteins called beta amyloid and transthyretin, and what happens is they are get produced outside of the various cells. Beta amyloid is in the brain, specifically what causes Alzheimer's, and transthyretin is actually related to cardiac conditions. And so these extracellular junk proteins accumulate externally of the cells. And so there's actually something interesting that we discussed. And so what happens is we can actually be vaccinated against these, and that's pretty cool. There's a company called ELAN, ELAN, that showed it can actually be removed in mice. And so that's pretty cool. Essentially, we can take all this plaque out and then let the immune cells gobble it up. And so the way we do it is by simply using antibodies against these proteins, against these extracellular proteins, right? And these are specifically kind of these extracellular proteins that accumulate, right, outside of the cell. We talked about glycation in the last video. That was more for like structural type of proteins. And these ones here accumulate, for example, beta amyloid accumulates in the dendrite and in the supporting cells of the nervous system. And what happens is that they prevent the functioning. So for example, in this case, in the brain, so you start to forget, etc. What leads to Alzheimer's? And and what happens is by using like a vaccine essentially and having like a booster shot, just like we were used to do when we were born, having a series of boosters will allow us to use these antibodies against these kind of extracellular um, plaque type of proteins. And so this is actually close to clinical studies. And also, so this is actually pretty interesting because what happens is um, it was actually able to target and stop various conditions. So not only beta amyloid, another study targeted a different antibody, and it completely eliminated other many conditions, because this antibody was similar to other antibodies which was responsible for other conditions. So by simply using an antibody against one condition, you have immune cells which are then gonna remove all this plaque, and that's gonna not only be helpful for one, but for several conditions that have antibodies responsible for the plaque that are very similar to that antibody that you're targeting. And so that's actually pretty cool. And so unfortunately, so it is close to clinical trials, but unfortunately, the people who used it had a lot of immune responses. And so they had reactions to them. And so what we can do then is the next step, and it's still in clinical trials, is to simply target a specific part of that beta amyloid to decrease the immune response, because the immune response can be against a specific portion of that beta of that amyloid protein. So this is really cool because amyloidosis disease, this disease that are caused by these plaques, usually target very old people. So it's like the elderly type of population. And so if there's nothing else that kills you, right, in the really old people, it's going to be this kind of extracellular junk. So it usually affects the really elderly, this specific sense type of target. So if you manage to live for a very long time, let's say being a centurion, it's this kind of thing that's usually responsible for ending people. And so that's why it's very important to discuss as well. And then the next type of category is called senescence, cell senescence. And these are cells that don't replicate, but they don't die either. They're just there. They never die. They're death resistant. And that's bad because these cells actually release a lot of molecules that facilitate the division and the progression of cancer. Because what happens is they release these epidermal growth factors to stimulate cell growth and division, as well as other factors which are enzymes that break down the matrix, which allows metastasis to happen because cells can go through their boundaries, as well as the promotion of new blood vessels to give cancer cells the food that they need to feed them, to give them the nutrition. And so they actually promote a helpful environment for cancer cells. And so it's very important to remove them. And we can do them by you looking at specific markers that these have. So work has been done already on immune system cells. There are these anergic T cells that actually actually also kind of like stop dividing, but they also crowd up all the other cells. So we can't mount an effective immune response because these anergic T cells take up space from all the other immune cells, from all the other T cells and B cells and whatever. And so it's very important to do that. And so actually what we can do is there is a protein called BCL2 that prevents these anergic cells from committing apoptosis or programmed cell death. And so but there's this antisense RNA against this BCL2 gene that can stop these anergic proteins and then allow more room for the other immune response. So we can essentially rejuvenate our immune response. So this is an example of these kinds of cells, these anergic T cells that are just sitting there and prevent the immune cells from being active. Another example is cancer cells, and we can actually target them too by using a promoter, right? And the same thing for the anergic T cells. We can use a promoter so that these genes only go to these anergic cells because we don't want them to go everywhere else. So for cancer cells, we can use a telomerase promoter because only cancer cells have telomerase. It's a specific enzyme that only cancer cells have. And so by doing that, we can actually target them. Another example, there's actually a bunch of them. For example, you can use epidermal growth factor promoter to target neuroblastomas, to target brain cancers, because these cells have this epidermal growth factor receptor, and so we can use kind of this to target it. And so there's many ways you ensure that you have a targeted selection so it doesn't go to all the healthy cells in the body and mounts a crazy immune response against you. So that's very, very important. And so what happens is you can actually use a promoter for cancer cells by attaching it to another toxic substance. And so this there's actually a drug that is gonna find the cancer cells using telomerase or any other type of promoter. And what it does, this kind of drug, is it binds to a certain protein found in cancer cells. In fact, in other cells too, but it only will go to the cancer cells because of the promoter which you targeted, which we use to target cancer cells. And this type of protein binds to another protein called thymine kinase. And what it does, it sticks itself into the DNA. So I think it's called ganciclovir is the drug. And so what it does is it prevents the replication of cancer cells. So we essentially put a standstill. We halt their replication by simply putting this drug, ganciclovir, in place of thymine when cancer cells replicate. And so they can't put any more nucleotides in there. They stop their division and they just die. And so that's kind of but again, we want to use a telomerase promoter or a drug to only target the cancer cells, these particular cancer cells, and not in any other healthy cell in the body. And so this is actually really useful. We can do it both in terms of a drug or in terms of gene therapy, and we can target the cells that we want to, right? For example, anergic T cells, which crowd up our immune system when we get older, as well as cancer cells, both of which are usually age-related type of situations. And so for senescent cells, they have distinct markers. These are cells that stop replicating, but they actually promote cancer. And so we can use similar approaches that we use by simply using a promoter or a suicide gene, as it's called, to only target the senescent cells, right? We can use things that are called light therapy, BNCT, involving boron and neutrons, right, which are very toxic because they can kill the cells. But again, whether we use light therapy, BNCT, dendritometers, I think they're called, we want to have a promoter to target, as well as this toxic mechanism to kill the cell. Well, and so in the case of cells senescence, again, we want to target these cells because they're bad. They can cause cancer, and they have distinct markers like SA beta gel. And there's a person called Judy Campsini at Berkeley who's working on this right now. But there's actually something called SA beta gel that they release, and that's how you know it's a senescent cell. There's also ways to double check mechanisms. There's other proteins as markers that senescent cells have, like P16, but they're not as reliable as this SA beta gel, unfortunately. So this is probably the best one to kind of use against them. And so we can simply insert the gene therapy, a suicide gene, right? Or we can use any of the other techniques, and kind of we can target those cells using the markers that they have. And so that's a really, really cool approach. So we can completely remove this cell senescence, because what happens as we get older, our cells stop dividing. So this is another very important sense category that I'd like to mention. In the next video, I will discuss the last sense category, which is extremely important, and that's the our deletion of cells. So here, kind of, I talked about what happens when these cells go kind of like haywire, when cancer cells or these allergic T cells, right, they just completely crowd up the immune system, and they just don't disappear, or cell senescence, they just don't disappear. Well, in the next video, I'll talk about the deletion of cells. And as we age, we have less and less cells. And so what can we do? And I will specifically reference stem cells when I discuss this sense category. I hope you enjoyed this video. If you haven't, please subscribe to my channel, and I will see you in the next one. Ciao.